On April 14, 2005, H.R. McDaniel, MD, traveled to Denver, Colorado to speak at the Capitol building as an invited guest of State Senator Sean Mitchell. Dr. McDaniel spoke for elected officials, your staff members, and interested guests in Senate Hearing Room number 356 at 9 a.m. Dr. McDaniel is a graduate of the University of Texas Southwestern Medical School and has spent 20 years practicing anatomical and clinical pathology, including positions as the Director of Pathology and Laboratories and Director of Medical Education at Dallas-Fort Worth Medical Center. In 1981, he began research at Fisher Institute for Medical Research using a bean extract to stimulate the immune system. In 1985, he added the work of scientists that had isolated the active principle of the aloe vera plant and conducted the first government-monitored studies in humans using this glyconutrient, aloe polymannan. Ladies and gentlemen, Dr. Reg McDaniel.
Now, we would not be talking today if in 1994 the Dietary Supplement and Health Education Act, referred to DSHEA, was passed. And it had in the preamble a statement that is law of the land that I can assure you I did not learn in medical school. It says science has shown that optimal nutrition is essential for good health, to prevent disease, and to restore health. That is a very vast-reaching statement. Hopefully to hook you into the power of this. Now, having read that, you might say, well, that just kind of goes along with common sense of clean air, clean water, good food, exercise, sleep. But the power of optimized nutrition exceeds anything I ever thought was possible, dreamed was possible, or taught others was possible. This is Brooke, that was born with about a 3-centimeter length blind pocket for an esophagus. That's incompatible with life, and she was sustained by hyperalimentation. But the wonders of modern medicine, they took a segment of small bowel and tunneled under the sternum and brought it up and hooked her stomach to that blind pouch to be able to get food in. As you might imagine, that doesn't work too well. And she had many studies over a period of time, but the mother got her on the dietary supplements we're speaking of, and as they did studies, they realized that down at the stomach, a protrusion began to come and began to grow upward, like a esophagus does in the embryo, and the blind pouch began to extend. The reason I show you that is I hope you'll say, how in the world is this possible? Like this one, Andrea, in California, at 25 weeks when they were doing scans of the uterus, ultrasound, they realized that the arms and legs were not proportional to the body and the head that they should be. So they did amniocentesis and found out she had the gene defect of achondroplastic dwarfism. You're aware of that. It's Snow White and the Seven Dwarfs. And this is considered a permanent, forever and always condition. In pregnancy, the mother started these dietary supplements added to her prenatals. And she was born and she had the deformities of the head and the very short from the wrist to the elbow proportions of the long bones, and immediately started adding the dietary supplements to her diet. She is now 5.5 years old, and the doctors are astounded. Her face changes, the proportions have normalized. The lengths of her long bones are normal, and they've x-rayed the growth centers, and rather than being disorganized, now they are normal, and they anticipate normal development of this child. I hope your question is, how in the world is this possible? Well, for a long time, fact is, we find out that it was Aristotle, over 2,000 years ago, that first wrote, that we have evidence of it, that the salamander can have its tail cut off or one of its legs, and it'll grow it back. In 1779 in Italy, a biologist cut the eye out of a salamander and said within a year it grew its eye back with the lens. And a Frenchman, Binet, said, wouldn't it be wonderful if we could figure out how to do this in human beings, or something along this line. And what we've been seeing really did not hit me with the full impact of the significance of some things we've been seeing until I read the book, The Proteus Effect, by Ann Parsons, a writer who for nearly 200 years research has been done trying to figure out how to do what the salamander does in humans. We have not grown any new legs, and of course we don't need a tail. We have fallen into, by accident or divine intervention, and it started for me in 1985 as a member of a group of scientists of over 50, MDs, PhDs, veterinarians, master's degrees, graduate students, and others. Why have human beings been using aloe vera for over 5,000 years to promote health? In many primitive languages, and it's been used every continent, every civilization up to the modern era. The language for this plant has been the medicine plant, the healing plant, the treatment plant, the miracle plant. And the analytical chemist in our team fractionated the gel from the leaf and found at the Fisher Institute for Medical Research a fraction that had antiviral, anti-tumor, and health-promoting effects, and it was a molecule called mannose. Most of you are familiar with the sugar glucose. It's the culprit in being too high in diabetes, though it's essential to health. We all have to have a blood glucose. But this is very similar in structure, but it's a different sugar, and it's used more in synthesis of glycoproteins and glycolipids, cell or body structure function molecules than for energy. When this was presented to scientists and medical people, no one wanted to be seen talking to us, because science and medicine had long ago decided that sugars were simply burned for fuel, energy to operate the body, and that important functions in the body were handled by genes or nucleic acids, proteins, which are chains of amino acids, and hormones. A very key paper was published in 1985 by Kornfeld, husband-wife biochemist team from Washington University at St. Louis. And it is 20 miles of difficult, complex biochemistry. And with a graphic artist, we have kind of summarized, and I will not go into the great detail of what is in this graph to summarize that paper, but fundamentally there is a model of a cell, and we have trillions of cells in our body. And this is what you can see at about a 500 magnification under a microscope. A nucleus with the genes and the nucleic acids, which are the programs that a sperm and an egg can come together and build a human body and operate it. The instructions are there for defense, repair, healing, regeneration, and to keep things in balance within the cell. At this magnification, it looks like there's nothing in the cell out here, what is called cytoplasm. But under electron microscopy at 100,000 magnification, there's an assembly line that makes all the structure function molecules in our body. And you can understand this if you can understand how a major automobile manufacturer makes vehicles. They bring in parts, they have a design. If you have all the parts, you hook them together right, it looks right, and it functions. And that's what each cell in our body does. In this paper, a key thing was present, and this is the molecular depiction of what is going on this assembly line. In the second step, right here, 9 molecules of mannose 6-phosphate are added to amino acids. If you recall, I said the active ingredient from the aloe plant was mannose. This is why human beings have been using aloe vera for over 5,000 years, and this we recognize is why we were seeing such incredible things. Because when we added the mannose from the aloe plant in concentration back to the modern diet, we had parents, patients, doctors, nurses saying, look what happened. It's a miracle. I'm here to tell you it's no miracle. No more a miracle than people used to die of scurvy, pellagra, and beriberi. Over 2 million British sailors died of scurvy alone. I've been a physician 42 years this year, and I have never seen a case of scurvy, pellagra, or beriberi, because we found out substances so small it's difficult to even analyze them are vitally important. And if they're missing from our food, it can be lethal and cause deformity. And that is the nature of this. Another point about this is we raise mannose sugars in the rice paddies and the grain fields across North America by the untold tons. But when you make white rice and white enriched flour, you strip it out, put it into animal food, and when you add it back, incredible things happen. Now, having found this before the DSHEA law was passed in '94, we entered into a new drug approval process, which is in 3 phases. First, it's toxicity phases, starting in animals and tissue culture. We did this with the active ingredient from the glyconutrient now, glyco from the Greek, meaning sweet or sugar. And our problem was we found no toxicity. That is medical heresy. The father of pharmacology, Paracelsus, 500 years ago, said all drugs are poisons. The benefit depends on the dose. And to not be able to, in huge amounts for extended period, to cause any toxicity to the liver, kidneys, bone marrow is without precedent. Now you go into a second phase, a third phase, and the point I want to make is that it takes about 10 to 20 years to go through this, and the U.S. pharmaceutical industry says it costs about $840 million to do this. I saw in Wall Street Journal that Vioxx, they claim $813 million was spent getting one claim for pain for arthritis. Well, fortunately, with the Dietary Supplement Act of '94, we abandoned this because dietary chains of mannose sugars. That doesn't sound like a drug, does it? And it's not toxic. And in fact, it doesn't work like a drug. But a key point of this is why new drugs cost so much. And if we had been successful in going through this, I can assure you, what is made available for 65, 75 cents a tablet or capsule or in bulk for 25, 35 cents would be selling somewhere between $8 and $15 for the same amount. Well, at this point, we abandoned it because this law was passed. Along the way, we had been seeing some unusual and unexpected clinical responses. Fact is, responses that I was taught and taught others will not happen. We had Down syndrome children getting out of special education, beginning to remember and be healthy and even change their appearance. We had children with gene defects where their brains don't develop, leukodystrophies, come out of coma and begin to develop. We had Alzheimer's patients that had been mute for 1-2 years, now can stand before you and talk to you just as I can. We had old stable strokes up to 26 years, major motor and sensory defects return. We had autistic children and Asperger's, a mild form of this, and dystonias and ADD, ADHD, Parkinsonism. We have over 300 documented Parkinson patients that have responded. Huntington's chorea, traumatic brain injury, and then fetal alcohol syndrome, and I have a asterisk there because I was invited to go to Canada to explain how these children had gone from special education, IQs less than 50, to regular classes and estimated IQ over 100. This was kind of like a slap in the face and a wake-up call that, as I showed you the assembly line of just restoring a few missing parts, just did not seem to cover the significance of this. And there was a clue back when we were doing the phase 1 toxicity studies before the DSHEA Act that after 1 week of adding this to the diet of dogs in their peripheral blood, and it was followed then by AIDS patients, ulcerative colitis patients, cancer patients, then hospital employees, students, on rotations through the hospital, in other words, normals. And for orientation, this is the normal size of a red cell. These are platelets, 1 to 2 micron, about 6 micron here. These are lymphocytes, the nucleus about 6 microns. But in the peripheral blood, there were huge cells. And here's one that is, the gene is turned on and it's begun to make substances that are structure function. For over a year, we call these cells G cells because that's what I said the first time I saw them. Gee, I've never seen anything like this in blood. But this precipitated studies to determine could this be toxicity or damage. And eventually we found out by sophisticated instrumentation that these cells were monocyte macrophages which participate in defense, repair, and healing, which we were seeing clinically. And so we said, well, these are just very active cells doing what they always do better. If you will notice, before taking the products, if you had a white cell count of, say, 10,000, there would be 10,000 dots here. But there's a fluorescent channel to identify what kind of cells these are with an antibody with a fluorescent tag on it. And most people are aware that AIDS patients have a virus that attacks their CD4 helper lymphocytes. And if you had the proper antibody with the fluorescence on it, the computer can separate it out and you can get your count of how these cells have been attacked by the virus. So these could be CD4 helper lymphocytes. Under the red arrow, there are no big cells. But a week after adding to the diet the active ingredient from the aloe plant, the polymannose, here are the big cells. And with the proper antibody, they are monocyte macrophages, and we thought we had the answer to the problem and forgot it for 15 years. Now, I showed you an assembly line, and a very key point I wanted to make from that these are substances that are made on the assembly line, and they're called interleukins, tumor necrosing factor, interferons. Here are colony growth factors for repair. But these are mixed leukocyte cultures overnight. And in standard media is the solid bar. Then you add 1, 10, 100, and 1,000, and you see the synthesis of these defense, repair, and regeneration molecules go up as the supply is increased. These are involved with communication between white cells, particularly, that I have found in the lung a virus or in the kidney a bacteria or an abnormal cell, and to do something about it. Remember, they're called cytokines, cell activators. They're sent between cells much like IBM cards. And on the assembly line I showed you, as you add more of the mannose, more is made, and this allows increased promotion of health. Now, a paper was published in the National Academy of Science 2003, and it's entitled A Human Peripheral Blood Monocyte. Remember, that's what we call those cells, CD14. Derived subset acts as pleural potential stem cells. Pleural potential stem cells means from your own bone marrow, adult stem cells can make all the cell layers that embryonal stem cells make, from the surface ectoderm, the cells in the middle, the mesoderm, the cells at the center, the endoderm, which are epithelial cells on the surface, endothelial cells that line the capillaries of the blood, liver cells, T lymphocytes, macrophages, mature ones, and neurons. Remember the list of the conditions I said that were responding that should not respond. Well, this shows that the type of cells that we were showing on the flow cytometer can make all of these, and they're listed in the paper as you add cytokines, the very molecules that I just showed you in the bar graphs that your own cells can make. They were using recombinant DNA and other exogenous added to the stem cells and getting this differentiation. Now, to show you the magnitude of the fetal alcohol syndrome children from the Indian reservations in Canada, these are 36 traits. They're questions that are asked of the school-age fetal alcohol syndrome child. One of the questions would be they do things in public that is only appropriate to do and private, and you can't teach them otherwise. And it goes down through many questions like this. And over time, they have learned that if you have of these 36, from 10 to 15 yeses, these children will never be educatable, trainable, employable, never live independently, and be a ward of the state for life. These 7 children averaged 27.7. The lowest was 20. That is terrible fetal alcohol syndrome damage from the toxicity of alcohol. But the adoptive and foster parents said, on putting them on these supplements that are designed for children, they've improved. So we modified this and said if you said yes on a score of 1 to 10 from normal to terrible to impossible, how bad it is. And it was an average of 8. Terrible, terrible fetal alcohol syndrome damage. But after a minimum of a year on the supplements, they averaged 2.5, and that's a 67% improvement. Now, that's just numbers. Let me translate that into living. This little girl at the age of 9 was still in special education and had not been able to master her numbers or her alphabet, much less read or add or subtract, and was a behavior problem at home and in school. She has been on these products 3 years. She's now in regular classes, making A's, B's, and C's, writes poetry insightful of her experiences that thrills her teachers and foster mother. But she called us excited that she had gotten her hands on a new Harry Potter book of over 800 pages and read it in less than a week for pleasure and then could tell you about it. That is what I said brought a crisis in this. Something beyond what we'd done before had to be happening. Here is also an example of a study that has started in Houston, Texas. The first 4 patients with Alzheimer's disease, using the Alzheimer's Global Score for the association, at 90 days by adding the dietary supplements, they had a 42% improvement in their scores on their Alzheimer's. I understand now they have about 50, and I'm looking forward to analyzing those. But we've had others that have done incredible. Well, while I was making assessments of the fetal alcohol children and the inadequacy to go up there and explain the degree and the nature of changes, this paper was published in the Journal of the American Medical Association as a summary of a paper that had been in the proceedings of the National Academy of Science. 2 science authors, MD, PhD, Eva Mezzi from the National Institutes of Health, and Dr. Barbara Crane, neuropathologist at Johns Hopkins, report in this on 4 female leukemic patients at Johns Hopkins, 2 teenagers and 2 in the 20s, that had all the treatment for leukemia, including high-dose chemotherapy with bone marrow rescue from only male donors. It did not work. They died. They came to autopsy, and they analyzed their brains, and they found out that in the brains of all of these females were multiple neurons. This is under ultraviolet microscopy light. Here's the axon, the neuron, the highest functioning cell in the human body where information, relationships, values are made, control of our body is centered. But they found little red fluorescent dots because they had treated these neurons with an antibody with a fluorescent tag against the Y chromosome. Only male cells in their genome have Y chromosomes. And here we have sitting in a female brain that had had bone marrow transplant from males only, Y chromosomes. And as Dr. Mezzi said, it was like there was a signal in the brain. We've lost a neuron. Come here, little stem cell, migrate into the brain and become and replace this neuron. This is absolute proof of this. And when we saw this, and then we went back and found the paper that showed the transformation of adult stem cells by the Chinese investigator Zhang, we knew that this population of cells that we had seen was the cause or the source. Now, to summarize this, The dietary supplementation supports the development of patients' adult stem cells. There's migration and transformation and differentiation under the patient's gene control, and it seeks out damaged cells by using normal apoptosis. That is normal physiology that goes very slowly on in our bodies. But stem cells from our own bone marrow migrate in and replace damaged and aged cells. The supplementation enhances this. Very important, there's no rejection of foreign cells, has been noted in type 1 diabetic stem cells from donors and embryos that were celebrated that they had infused. They migrated into the pancreas of the diabetics, transformed into beta cells, and began to make insulin, and the individuals no longer needed insulin for about 8 to 10 months, and then it went backwards because on the membrane of those cells is just when you have an organ transplant for a lung, heart, kidney, that it's someone else, and the immune system recognizes it and destroys these valuable cells. This is done at a cost of $25 to $50 per week. Hundreds of patients demonstrate that